In this video, we're going to focus on the regulation of gene expression in prokaryotic cells. But before we get into the nitty-gritty details of molecular mechanism and how regulated gene expression occurs, I just want to spend a moment thinking about this issue from a larger perspective, from the context of regulation of cellular metabolism and function. We've talked about enzyme regulation before in the class, and we've discussed examples of metabolic pathways in which feedback inhibition occurs, such that the product of a metabolic pathway can inhibit an enzyme in the pathway and effectively shut off its own production. One of the examples we saw was the pathway responsible for synthesizing the amino acid tryptophan. There are 5 different proteins needed for this synthesis, and if tryptophan concentrations rise in the cell, the molecule acts as an allosteric inhibitor of an early enzyme in the pathway, which stops the cell from continuing to synthesize more tryptophan when sufficient tryptophan already exists. And we talked about the logic of that regulation. It allows the cell to conserve energy and respond in a very dynamic fashion to cellular needs by shifting the use of cellular resources where they are most needed. But this type of feedback regulation can actually operate even upstream of the enzymes themselves, at the level of the genes that encode the enzymes. In this case, we're talking about regulation of gene expression. If the cell has sufficient tryptophan, then not only does it not need to waste cellular resources making more, but it doesn't need to waste cellular resources and energy synthesizing the enzymes required to make the tryptophan. So both of these types of regulation occur simultaneously in cells. And it's this type of regulation of gene expression that's the focus of this video. The example that we're going to be looking at is one of the first gene expression regulation mechanisms that was ever worked out. This is work that was done by a couple of French scientists, François Jacob and Jacques Monod, and they shared the Nobel Prize in 1964 for their discoveries because they were so seminal to our understanding of how gene expression can be regulated. Many of the basic principles that they figured out in prokaryotic systems are very applicable to what we see happening in eukaryotic cells as well, as we'll see when we talk about eukaryotic gene regulation. They studied gene regulation in a bacterial species called Bacillus subtilis, and they were trying to understand the molecular basis for a phenomenon observed in bacteria in which if you are growing bacterial cultures in the presence of more than one sugar, let's say glucose and some other sugar, lactose, mannose, arabinose, whatever, you see this very characteristic growth curve that's referred to as dioxy, which is this sort of biphasic curve. This shows the growth in the number of cells in a bacterial culture over time. On the y-axis is shown the size of the population or percentage of cells, and on the x-axis is time. What's observed is this biphasic curve where we see an exponential increase in bacterial growth and then a leveling off or stationary phase where no further expansion of the population occurs, followed by a second exponential increase in the population and then a second stationary phase and leveling off. If we followed this out even further, as the nutrients are depleted in the culture, we'd see this culture population kind of crashing out and cells dying. But this two-phase growth curve is very characteristic of providing bacteria a preferred sugar, which is almost always glucose, and some other sugar that they are less able to efficiently extract energy from. If this were, for example, lactose, which is depicted over here on the right, we would see exactly this same kind of curve. So what's the explanation for this? Well, it turns out that what's happening here is that the bacteria are utilizing their preferred sugar, glucose, first. And only when the glucose is completely used up do the cells shift their metabolism to be able to utilize the lactose sugar. Now, it's not completely understood why they do this. Since lactose is made up of glucose and galactose, it's just about as easy to use. But presumably there's some energetic advantage to utilizing the glucose first. And what's happening at this lag that we see between the two growth phases is that the bacteria are shifting their metabolism and activating expression of the genes that are required to metabolize lactose. So the first thing to recognize is that the default state for these lactose metabolism genes is off. The cell doesn't make the enzymes and other proteins needed to metabolize lactose if there's no lactose around. That makes sense. Why waste that cellular energy making enzymes to break down lactose if there's no lactose around? So question number 1 is, how do the genes know whether there's lactose around or not? What's the specific molecular signal keeping expression of the genes off when there's no lactose around? The second thing to recognize is that even if lactose is around, if glucose is also around, the genes will stay switched off. The cell will preferentially use up all of the available glucose before switching on these genes to metabolize lactose. So question number 2 is, how is that information communicated at the molecular level? How is it that even in the presence of lactose, the genes stay switched off if glucose is still around? These two conditions both have to be met in order for us to see the activation of these genes associated with lactose metabolism. Lactose must be present and glucose must be absent. And this is what Jacob and Monod figured out. What exactly is happening at the level of the DNA that allows this metabolic switch to occur? So what did they discover? Let's take a closer look at the genetic instructions for those lactose metabolism genes and the regulatory elements that control whether they are expressed or not. We've talked before in the class about prokaryotic operons, which are clustered sets of genes that are functionally related to one another and coordinately expressed, meaning they're all transcribed and regulated as a unit, either all expressed or all not expressed. The genes for lactose metabolism are present within this type of cluster, the lactose or lac operon. There are 3 protein-coding genes in the operon, called LAC-Z, LAC-Y, and LAC-A, all under control of a single transcriptional start site, indicated by the arrow here. All 3 of these genes are transcribed as one unit, resulting in a polycistronic mRNA that contains the protein code for these 3 separate proteins, with separate start codons to translate each one separately. LAC-Z encodes an enzyme called beta-galactosidase, which is the enzyme that cleaves the lactose disaccharide into glucose and galactose monosaccharides, after which the cell can use these monosaccharides for glycolysis. This beta-gal enzyme also catalyzes a side reaction, isomerizing lactose into its stereoisomer, allolactose, the importance of which we'll see in just a moment. LAC-Y encodes a membrane transport protein called lac permease, which is responsible for transporting lactose into the cell. Without this protein, the cell can't obtain lactose from its environment. LAC-A is a bit of a mystery. It's known to be a transacetylase enzyme, but its importance for lactose metabolism isn't understood. It seems to be dispensable, so we'll ignore it for the purposes of our discussion. Upstream of the transcriptional start site are the DNA sequences that are required to appropriately transcribe these genes. P is the promoter sequence, which, as you've previously learned, is the binding site for RNA polymerase. Directly adjacent to the promoter are 2 types of regulatory sequences that are going to be important for repressing or activating transcription in response to the presence or absence of lactose and glucose. First, there are the operator sequences, symbolized by the O, and there's a cap protein binding site, or CBS. So the cap binding site, operators, and promoter are sequences in the DNA to which RNA polymerase and regulatory proteins bind to control transcription of the LAC-Z, Y, and A structural genes, which encode proteins needed for lactose metabolism. So what are these operator and cap binding site sequences? The operator is a binding site for a repressor protein. This protein negatively regulates transcription of the operon. When repressor protein binds, it blocks RNA polymerase from being able to transcribe this set of genes. Notice that there are 2 operator sequences that flank the promoter. Repressor binds as a dimer. In other words, 2 copies of the repressor protein bind, one on each operator sequence, and they also bind to each other, causing the DNA to form this loop, which prevents access to the promoter by RNA polymerase. This repressor protein is actually encoded by a separate gene in the genome, so it's not part of the operon. This is called the LAC-I gene. It has its own promoter, it's always expressed, and it's always going to be producing this repressor protein that can sit down and bind here to the operator sequences, and when it's bound, it's going to block RNA polymerase from binding to the promoter and being able to transcribe. Now, the cool thing about this is that the repressor protein is itself negatively regulated by allolactose, the isomer of lactose. So when lactose is around, it's isomerized to allolactose, which binds to the repressor protein and induces a shape change in the protein that prevents repressor from binding to the operator sequence. So if repressor is not bound, this looping out of the DNA doesn't happen, and nothing is preventing RNA polymerase from having access to the promoter. So this is the molecular mechanism by which this LAC operon senses the presence or absence of lactose. When lactose is absent, repressor is bound, repressing transcription. And when lactose is present, repressor is not bound, so it's not inhibiting transcription. But as it turns out, even when lactose is present, transcription of this operon only occurs at very low levels if glucose is still around. So why is that? If we take a look at the promoter sequence of the LAC operon and compare it to the ideal promoter sequence for the bacterial RNA polymerase, what we see is that the promoter sequence isn't an exact match. The ideal consensus sequence in E. coli has a TTG-ACA sequence, about 35 nucleotides upstream of the transcriptional start site. And another important sequence at about -10, TAT-AAT. But if we compare the LAC operon promoter sequence to that, we see that it doesn't exactly match. At -35, the sequence is TTT-ACA, and at -10, the sequence is TAT-GTT. So the LAC operon has a promoter that doesn't efficiently recruit RNA polymerase. It's a so-called weak promoter. So without help, RNA polymerase can bind this promoter, but not very effectively. It tends to fall off. In order to bind tightly and be able to effectively initiate transcription at high levels, RNA polymerase requires an activator protein, which is the CAP protein. CAP stands for catabolite gene activator protein. The CAP binding site is directly adjacent to the promoter, and when CAP is bound, RNA polymerase is stabilized on the weak promoter and is able to transcribe. Now, whether CAP is bound or not depends on whether glucose is still around or not. CAP is the molecular sensor for the presence or absence of glucose. So how does that work? CAP is allosterically activated by cyclic AMP binding. Remember cyclic AMP from our signaling unit? It's the same one. Just like more complex cells, bacteria have adenylyl cyclase enzymes that convert ATP to cyclic AMP, and they use cyclic AMP for all sorts of regulatory and signaling functions. This is just one of many. CAP can't bind to the CAP binding site unless it has cyclic AMP bound to it. Cyclic AMP binding induces a shape change in the protein that allows it to recognize and bind the CAP binding site. As it turns out, glucose inhibits adenylyl cyclase. So as long as glucose is around, cyclic AMP won't be made. It won't bind to CAP. CAP can't stabilize RNA polymerase on the LAC operon promoter, and transcription of the operon can only occur at very low levels, if at all. When glucose is no longer around, adenylyl cyclase is no longer inhibited. It synthesizes cyclic AMP, which allosterically activates CAP protein, which binds to the CAP binding site, stabilizing RNA polymerase so it can transcribe the operon at high levels, but again, only if repressor protein is not bound. So to activate full high-level transcription of the LAC operon, lactose must be present so that allolactose allosterically inhibits the repressor from binding to the operator, and glucose must be absent so that cyclic AMP binds to CAP and activates it, which allows it to stabilize RNA polymerase on the promoter. Only under these two conditions will we see high-level transcription resulting in the synthesis of lots of mRNA from this operon and translation of lots of beta-gal enzyme and lac permease transport protein. Now, to be completely accurate, it's not glucose itself that directly inhibits adenylyl cyclase. Rather, it's glucose transport into the cell that ends up indirectly inhibiting adenylyl cyclase via an enzyme called E2A. As glucose is transported into the cell via its transport protein, it gets phosphorylated by E2A, which loses a phosphate in the reaction. So essentially, a phosphate group is transferred from E2A to glucose. If glucose is not being transported into the cell, then most of the pool of E2A enzymes in the cell remain in their phosphorylated state. And in this state, this enzyme activates adenylyl cyclase. But if glucose is being transported into the cell, then most of the E2A pool is dephosphorylated. And in this form, it doesn't activate adenylyl cyclase, so little cyclic AMP is made. So it's not glucose directly, but glucose transport indirectly that is influencing levels of cyclic AMP in the cell. Another thing I should mention for full disclosure is that this CAP sensor is only part of the story in terms of glucose regulation. Because when glucose is being actively transported into the cell, and this E2A enzyme pool is mostly in its dephosphorylated state, E2A inhibits the lac permease protein from transporting lactose into the cell. So in other words, as long as glucose is being transported into the cell, transport of alternate energy sources like lactose is inhibited. So if no lactose is being brought into the cell, then no allolactose is being made, which means repressor won't be inhibited. So in a way, glucose is also indirectly determining whether repressor protein is going to bind to the lac operon as well. One final thing to note is that this regulatory mechanism via CAP is commonly used in bacteria to regulate a whole host of operons with genes for metabolizing alternate sugars. It's used for the arabinose operon, the mannose operon, etc. So there's a common glucose sensing mechanism that's signaling to all these operons the availability of glucose or lack thereof, and potentially activating their transcription only when this preferred sugar is in short supply. To challenge your understanding of this regulatory mechanism, practice with different scenarios and predict how the presence or absence of these sugars will influence lactose transport, repressor binding, and CAP binding, and predict whether high-level transcription will be observed. We'll talk more about these in class.